So should I go ahead and start?
Yes, please.
Okay. Okay, so welcome everybody back today to the second day of the 3-day seminars on clinical trials. So today I'm going to be talking about some more design aspects of the study and conduct aspects of the study. And but as you see by the parenthesis, we're going to be talking about ethical issues. And as I said yesterday, almost everything that is done in clinical trials and the way we do things is dictated by ethical considerations because we're intervening on human beings. So stages of a clinical trial are fairly long process. It starts from, proposing to do it and getting fundings, getting some approvals from regulatory agencies. And before you start recruiting the first patient, you have to develop all the quality control processes, the study materials, the data entry and management systems have to be made in place. Then there's a process of recruitment, and intervention starts usually while recruitment is maybe going on still. There is also follow-up. As soon as you start recruiting somebody, you're following them up. So these three things are intertwined. And then when study ends, we definitely have a definitive analysis after all the data are cleaned up, et cetera, and then at that point. Now, conducting a clinical trial is something that is very important to be done with high quality. The purpose of a clinical trial during the conduct phase is to conduct a study to yield high quality data, okay? There are guidelines on this. We refer to these as the good clinical practices, and there's also good data management practices. And we'll talk a little bit more about what these involve. It always, when you're reporting this, you have to be transparent in what you've done. And my strong advice, if you're ever involved in conducting a trial, is to document all your procedures, because you may be called up to be audited, and in which case you have to be able to show that you have not committed any fraudulent aspects of the data and that you have good quality. And we'll talk also a little bit more about that. So what are good clinical practices? So what used to happen is that every country worked on its own, and they would develop guidelines for safety, quality, and efficacy of their pharmaceutical products. And so there's regulations, guidelines were different from every country. And this is what partially contributed to the high cost of research and development and high cost of the consumer afterwards, right? And so in 1997, so over now 25 years ago, close to, the governments of the United States, of the European Union, and Japan developed regulations that were common for the pharmaceutical markets among them. And why? Because what was happening is that maybe a drug was approved in Japan under their tight regulations, but it was not approved in the United States because it had not been done in the United States under United States guidelines. So this was an agreement among the regulatory authorities and the experts from the pharmaceutical industry. And then the Australia, Canada, the northern countries, and the WHO came up with this, what they call International Conference on Harmonization of Technical Requirements for Registration of Pharmaceuticals for Human Use. And all this is available on the internet for you to look into. So what did they do? They defined good clinical practices as generally accepted best practices for conducting clinical trials and device studies. It's not just for drugs, it's for any kind of clinical trials. They're defined as an international ethical and scientific standard for how to design a study, how to conduct it, how to record the information, and how to report trials. So it's very broad, encompassing, and involves the participations of human subjects. You have to be clear that these are not statutes or regulations, so they don't have the force of law, but basically they essentially do because they force people that if you have not done it that way, then the pharmaceutical regulatory, the regulatory authorities in different countries will not accept the results of a study. And so even though it's not law, it basically functions like a law. So what does compliance with the good clinical practices provide? It assures the people that the rights and safety of participants were protected, and that the data and reported results that arises is credible and accurate, okay? They have 13 principles of GCP that are key, and I will just briefly go over them, or quickly go over them, and you may notice, if you remember yesterday, that I said that the doctrines of clinical trials were set 1,000 years ago by Ibn Sina, and it's basically what GCP does. Okay, so these are nothing necessarily that new, but there's the Declaration of Helsinki for protection of human participants. You have to look at the risks and inconveniences beforehand. And so a trial should be initiated and continued only if the anticipated benefits justify the risks. And then the right safety of the participants has to be utmost importance, and non-clinical and clinical information should be adequate to support the trial. So there should be this equipoise, right? Clinical trial should be scientifically sound, described in a protocol, should be conducted in compliance with the protocol, and be approved by an institutional review board or ethical review board. The medical care and medical decisions is the responsibility of a qualified physician or dentist. Each individual involved in conducting the trial should be qualified. So this is talking about the investigators' qualifications to do and trained to do their respective tasks. Informed consent is an important process, and it should be freely given. So they call it free given informed consent, okay? From every participant prior to participation. The last ones are that the information and the data should be recorded and handled in the proper way. There should be confidentiality of the records, and that the investigational products that are used, for example, drugs, should be manufactured with applicable good manufacturing practice. And systems with procedures that assure the quality of every aspect of the trial should be implemented. So good clinical practices basically is addressing ethical reasons for doing the study well, but also it's protecting the integrity of the study by doing things with high quality, okay? So these are the rules or the principles. How do you implement these things? Well, if you go on their website, you can download the good clinical practices brochures and detailed processes. They provide guidance on key components of the trials. So what is the role and how to constitute an institutional review board? There's regulations about who is qualified to be an investigator. It talks about who is sponsoring things. Is it the pharmaceutical industry? Is it a contract research organization that is working along with the industry? All the details of what should be part of the protocols and protocol amendments. And then what's called the investigator's brochure and other essential documents. So all of the details of this is there. I'm not going to go into all of these, but wanted to mention that since part of the process here is basically to ensure high quality data and results are coming out, there's the mention of quality control and quality assurance. And many people don't see the distinction between those two words. And quality control is really the processes of monitoring. So they're implemented to improve the accuracy or timeliness of data or to meet some standards, while quality assurance is a systematic and independent assessment of the trial related activities and documents. So that's trying to determine whether the activities were conducted properly and the data were recorded, analyzed, and accurately reported according to the protocol or the sponsor's standard operating procedures, the good clinical practices, and the applicable regulatory requirements. So this is a difference between monitoring and audit. And so monitoring applies to everybody. When people do an audit, they usually will select a sample of subjects to check on them. Monitoring is a continuous process and it's part of the study. So that is an essential component. While an audit is something that can occur, not necessarily may happen. It is actually quite rare that it occurs. It's that if somebody suspects something, it may not be going well, or a regulatory agency sees that, oh, this is a very important study that can change practice, and we wanted to make sure that it's okay, they may impose an audit just to check it. Monitoring is done usually at all sites, and audit is at selected sites. Who is responsible? The monitoring is the sponsor that does it, and the audit is sometimes also the regulatory body's responsibility. And the monitoring is done as an ongoing thing by the site or the sponsor personnel. And an audit now is some independent body that shows up without telling you that they're going to show up and starts checking to make sure things are going okay. So when conducting a clinical trial, we said that you have to have some essential documents, okay? And one of those essential documents is the clinical trial protocol and amendments to it. And a protocol, as I mentioned yesterday, has to be registered, but it consists of what we would call a roadmap to assist investigators and site staff in the day-to-day management of the trial. So this is a place where you have all the information, a little bit of the background and the purpose and so forth, but it's mainly the procedures, the timelines, how safety is going to be protected, how data collection is going to be done, how data processing is going to be done, data analysis, and publication policy of the trial. It also includes who you're going to study very carefully. So there's some components to the protocol. You have basically some administrative information, but you basically give the background of what is being studied. You summarize other available past data, and then you give the study purpose and objectives. After you do that, you have the study design, which are the primary endpoints and the secondary endpoints, type of trial, the duration of subject participation, and aspects of statistical monitoring. And we'll talk a lot more about that tomorrow. There's also aspects about who is going to be included. So the subject eligibility requirements and how subjects will be treated. So what's going to happen if subjects want to withdraw? When will they be withdrawn? Will you replace them? Will you follow withdrawn subjects? All of these little details are part of this protocol. Mainly how safety and efficacy will be assessed. There's also a section on this necessary sample size, and we'll be talking more about that tomorrow, and the statistical analysis methods as well. Finally, you can see how extensive this is. This is not a little small 2-page document. This is a usually 20, 30, 50 pages or more document, right? It also deals with access to source data and documents, what agreements you have before the study starts of how you're going to get data, what are you going to do after the study ends, who's going to have access to the data. It talks about quality control, quality assurance processes, and ethical considerations, others like what is the informed consent process? What is the external monitoring for participant safety, the data safety monitoring board? I'll talk a little bit more today about that. All the data management documentation processes, and of course also the publication policy, if not addressed in a separate agreement. So it's very comprehensive about all of the processes that are going to be part of the clinical trial study, okay? And it is provided in some detail. However, it is not in extreme detail. So for example, they may mention what is going to happen and the timeline for subjects. So once they're randomized, they will have a visit, baseline screening visit, then they're randomized, then we'll collect data on week 1, on week 4, at 6 months, etc. This is the data that we're collecting. So it sort of outlines the study, but it doesn't say how you're going to measure that blood pressure or how you're going to measure a particular depression score. Those sorts of details actually will come in other documents like the manuals of operations and so forth, which go into real detail about how things are actually done, okay? The quality control processes are various, and if you want to get good quality, you have to do some of these processes. One of those is you standardize procedures. So you don't let people do things differently, especially if you're doing it in multiple centers. But even if you're doing it in a given center, you don't have people decide on a given day, oh, I'm going to measure weight this way, or I'm going to use this particular way of getting their cholesterol. You have to have the procedures standardized. You want to make sure that you get good assessments or quantifications of variances, right? Because when we start doing analysis, we really need to know these variances. So you have to look at, if you're doing repeated measures on people, we want to know it within person variability. But also if you are just measuring something once, what is the inherent variability within a person? Should I measure it twice? Should I measure it 15 times or only 3 times? People in the old days used to measure blood pressure 3 times and take the average, for example, because they realized there's a lot of within person variability on blood pressure. So you have to have a good understanding of sources of variability. You want to minimize measurement errors. So there's all these aspects of training people. You're calibrating machines and you train people. And actually, I didn't mention here, but you also then check people to see if their training was successful, right? And so we have a process sometimes called a certification, where you certify, oh, this person is able to do this task because they have been trained and they passed an exam that says that they are adequately trained. And even though they've done that, you still monitor their activities. You want to get accurate and complete data collection and accurate and complete data entry and processing. So now you're conducting a study, and where does quality come in when you're conducting the study? You want to look at the sources of data and the potential for variability due to the types of source of data. So for example, most clinical trials will involve a physician or healthcare worker doing a clinical examination. You have to train people to do it the way you want it done. You cannot let them do it the way they normally do it because everybody does it slightly differently. So as you standardize things, you realize that, oh, the clinical examination needs to be detailed as well, okay? Are we going to get data from devices? Like you're going to get a finger stick measure of cholesterol or of glucose, or are you going to do it from a laboratory? And are you going to have a central laboratory where all the bloods are sent to, and that one has been calibrated and has good internal and external quality control? Or are you going to have multiple laboratories that now you have to ensure that all the laboratories are comparable in what they're doing? Are you going to be using some other types of data that come from images or electrocardiograms or MRIs or CT scans, X-rays? Are you going to take verbal recordings of information? And then how are you going to analyze these things? Nowadays, many hospitals have electronic records. Are you going to be able to abstract that from them? Are you going to, for example, if you're doing a nutrition intervention, are you going to have patients have electronic diaries on their cell phones, for example? We did a study on gastrointestinal irritable bowel syndrome, and we had an app that was study developed for patients or subjects to record their experiences of pain and so forth electronically, right? So then you have all of these systems nowadays that people do. You have reading centers. So if you get an image, who is going to interpret this, right? So you have to have a machine do it, or you're going to have a person do it, who's going to be trained to do it. And finally, one of the most important aspects is, of course, the outcome of the study, and you want to assess it properly. And so sometimes, if it's not an easy thing to determine, we have endpoint classification committees, or sometimes I call them outcome adjudication committees. And they are very critical in determining whether somebody had the outcome or did not have the outcome. So the quality control procedures are particularly important there. And they depend on the study's data entry system. So there's all of these systems for quality control, but you start realizing that in most clinical trials, there are an enormous amount of information that is collected. And we as statisticians always insist, well, a second, if you're going to collect data and spend the money to collect it, you want the data to be of high quality, okay? But if you're collecting so many things, then the question is, can we get high quality on everything? So one of the things that statisticians tend to do during the planning stage is we try to advise the investigators, is it essential to collect that? Why are you wanting to collect this? How do you plan on using this in analysis? Or are you just doing it because, oh, once you have the subjects, it would be nice to collect such and such for maybe a future study? And so just to remember or keep in mind that when you collect more and more information, your resources, efforts, your time, your energy is now dissipated, and you're not necessarily going to be able to give the high quality to everything. So first of all, try to limit what you collect to essential things. But even those essential things are still quite a bit. And so now, when you're developing your quality control systems, you must decide what aspects of your study will essentially be critical and require being of high quality. Is it usually all? No. There's some things that are more important than others. And so the key aspects, those essential ones, have to be ones where you have good quality control systems, okay? Usually what is that? That's the eligibility criteria needs to be assessed properly, and the outcomes have to be assessed properly. Those are basically the two critical ones. Then comes a few others like the randomization, process, the intervention, administration, and so forth. But if you do a simple trial, at least the two things that are most critical are proper assessment and inclusion and exclusion criteria so that only eligible people are included, and that you measure the outcome properly. Okay. So the screening information is essential. The outcome variable is essential. And then you may want to consider also the clinical condition of the subject as essential. Okay. Now, when you collect the data, it is not as simple as we think it is. And we have not just simple questionnaires or paper forms that are being used. We have to do more complex types of data systems. Why is that? The reason we have to do more complex data systems is, and I call them data management systems, is because of the potential of needing to justify that the data has not been tampered with and that it is free of any potential manipulation or fraud. Okay. Why is this so? So it goes back to the ethics that we need to ensure that since this study can potentially change clinical practice or medical practice, therefore it was done properly. If somebody, doesn't matter whether it's from the study or outside the study or a regulatory agency or the government or a competing pharmaceutical company or something, if somebody alleges, I think this investigator did not do the study well, I think they may have tampered with the data. It looks too good to be true. Their result, I don't believe. If there is doubt that somebody makes up, and they can make it up for whatever reason, then you may be subjected to an audit. If you are subjected to an audit, this is very different from most situations where if you're accused of a crime, you're assumed innocent until proven guilty. Here it's the opposite. If somebody accuses you of having tampered with the data, you are assumed guilty until you prove that you have not tampered with the data. So in order to prove that, you have to have data systems that allow you to say, there is no way we could have tampered and we did not tamper. So you cannot just record things on Excel spreadsheets because people can make mistakes on that. You cannot do it on unprotected systems. You have to do it on protected system. And so the data management system is now a complex computer-based process that involves different things. So how you capture data, how you store the data, how you process the data, how you report or extract data from it, and all the features of security. So data capture may be on paper forms originally, but it has to be digitized at some point, or it can be done directly onto digital medium. And so there's, whenever it's digitized, there's a screen creation, there's validation, there's a field entry, calculated values. You don't let people calculate their BMIs, and interviewers calculate the BMIs. They give you the weight, they give you the height, and the computer calculates the values. How you handle missing values, all the notes or comments. Yeah, I measured this person's height, but the person was not feeling well and he was like this. I could not get him to stand straight. That's a comment that sometimes is also included in the data. How you check the data, you do reporting on it, how you extract it. There's the aspects of how you store it with sufficient capacity and the security. We have what's called an audit trail. And an audit trail is simply the system records, it's usually password protected. When somebody enters with their password, it records it. They know the exact time and date that person entered. It knows what that person did. And it said on this time, this day, this password entered the database. Some of them have privileges of browsing, some of them have privileges of editing and entering. And this person entered and went to this particular subject's data on this form and changed the value of this variable from this to this. And so that is kept up. That allows somebody to then trail and track who did what and how the data was monitored. So all of the systems make things very complex to do, but people do it. You have other features of randomization, query management, and coding. So these data management systems are usually part of the process, mainly driven by the potential of having to be transparent and that you have not tampered with the data. So there are then the Society for Clinical Data Management is a private organization, and they have developed what they call good clinical data management practices, sort of along the lines of the good clinical practices, but now they're focusing on the data aspects of the study. So it's things about the common types of errors people find are where the subject completes the questionnaire incorrectly, or provides incorrect or incomplete answers, or they don't follow the instructions, or there were not inadequate instructions given to the subject, or there's a site personnel have violated the protocol, or data was captured incorrectly on the source, or there's a transcription error, or human error in reading equipment or printout, a data entry error, and one of the huge problems that we'll talk about tomorrow, which is missing data. Okay, so these are types of common sources of error. There's also the electronic data acquisition error. Maybe there's a power glitch or something, the backup did not run, or data is linked to the wrong subject because the ID was mistyped or something. And there's a data clarification form or query, but it was not updated correctly. There's outliers, standard things, the outliers, inconsistencies, or there's some programming error or lost data. And in some cases, very, very few cases, the problem of fraud. Okay. So the GCP, as well as the clinical data management practices, are all of these dictates are based on ethics. And why do I keep insisting on ethics? So it's actually every design consideration, and the quality control systems are part of design considerations. But every design consideration is driven by ethical considerations. So even the selection of the study population, who to study, you don't study everybody, you study the population that is likely to benefit. It is unethical to start studying people that are not going to benefit from it. So why put them as guinea pigs if they're not going to benefit from it? The intervention arms that you're going to study have to be what we call clinically reasonable. So for example, is placebo ethical to do? And we talked a little bit about that yesterday. Should I do it or not? You have to have equipoise in the intervention arm. So the ones that I choose have to be equipoise. The follow-up procedures, many times we have things where it's really cumbersome. It's a burden to the subject to have to do these things. And so why force them to do it? If you have them do it all because I have to do it this way in the study, think about it. After the study ends, suppose you show that a very cumbersome follow-up procedure, like we had a study in ophthalmology where they had to put on eye drops for, they had keratitis, and they were using a cyclovir, and they were talking about at the design stage, oh, we should have them do 2 drops as soon as in each eye when they wake up in the morning, and then 3 hours later, then they do 1 drop in each eye every 3 hours, and then before they go to sleep, they do 1 drop. Who's going to do this normally? Suppose you show that this works in the study. You don't want to have something that nobody's going to follow after the study's ended, because humans don't follow very cumbersome practices like that. So also follow-up procedures, not only the interventions, but the follow-up procedures. Like, does the subject have to come every week to the clinic to be seen? Well, people are not going to do it. So you just have to be careful not to be a burden to the participants. The determination of sample size, we'll see tomorrow. For achieving certain power, you don't want to have an inadequately powered study. Otherwise, it's a waste of time. So you have to have necessary sample size. And as I've been mentioning, the data management and quality assurance procedures need to be done properly. The reason for all of these things are driven by ethical considerations is because it is totally unethical to conduct a poorly designed trial. And why is that? A poorly designed trial may end up with the wrong result. So it may be that the intervention is actually beneficial, but because you did a poorly designed trial, you're unable to show that. So now you are keeping a potentially beneficial intervention away from people that could benefit from it. So that's unethical. Or on the other hand, you could show in a poorly designed trial that something that is not of benefit actually is of benefit. And so now people will be using something that is worthless. So again, you're being unethical. So a poorly designed trial is considered an unethical practice, okay? So let's assume you've designed it properly, okay? So we're going to now move away from having designed things to conducting the study. When you're conducting the study, you may have designed things properly, but you still need to protect your study while it's happening from biases. And so we will talk about masking. We used to call it blinding, but some people say blinding. People that work in ophthalmology don't like the word blinding of studies. They like masking of studies. So this is more politically correct. And the assignment of people to interventions is called allocation. We want that to be objective, to be fair, and we use the process of randomization. Another thing that ethics forces investigators to do is to monitor safety issues during the conduct. What safety issues? Safety issues of the individual subjects. So the clinicians on a data safety monitoring board are actually monitoring the care of individual subjects, that they're not having adverse events, for example. But statisticians on the data safety monitoring board are looking at the group, at the collective safety issues, okay? So as we see, GCP dictates are based on ethics, and all aspects of designs of the trials are based on ethics. But let me talk a little bit more about this placebo. We had mentioned yesterday that it's sometimes unethical to do a placebo trial if there is something that already we know works. What is the implications of that statistically? So statistically, if we are having to, we usually have a control arm, right? If we are able to do a placebo control, then our new intervention is likely to show a large benefit, okay? But if we are forced for ethical reasons that we cannot use placebo because there is a usual care method that is adequate, that is being used, it is unethical to use placebo in that case. So now, instead of if the control or usual care has some effect, when I'm testing for superiority, the differences between the intervention and the usual care are going to be smaller than the differences between the new intervention and placebo. So therefore, it's going to be harder to show superiority. I'm going to need a larger sample size. It's going to be a more costly study. Aha, that's what happens. If I'm doing a non-inferiority, if the control has some effect, differences are smaller. And so it's actually easier to show non-inferiority. And so sometimes we are forced to have a margin that is even smaller than what we had expected. Right? So there's statistical implications of not being able to use the placebo as a control arm. So I'm going to, in the rest of today, talk about how some of the ethical design considerations, and I'm going to first focus on, and so they come in many places. You know, we talked a little bit about the control arm. The informed consent process is very important, but it's not really a place where statisticians are involved, so I'm not going to address that. We'll be talking tomorrow about the statistical power and sample size, and I'll talk today as well as tomorrow on the needs for external monitoring. But I want to talk about these processes here, the randomization and the blinding or masking. But let's first talk about blinding and masking. So what do we mean by blinding and masking? In the old days, people used to say, oh, we did single blind study or we did a double blind study. Nowadays, we are more careful about it, and we define blinding as who has knowledge of the intervention assignment. When we keep that knowledge away from those involved in the study, then we say this person is blinded to the intervention assignment. So it could be the assignment staff. Usually we say, participants. The participants need to not know what they're getting, but the staff, the clinicians, the study investigators also may need to not know what the person is getting. Do the study statisticians need to be blinded as to who's getting what? There's usually an external monitoring board. Do they need to be blinded as to who's getting what? Are the outcome assessors? So sometimes in some studies, it's impossible to mask or blind who gets what, especially if it's a nutrition study or a behavioral study like smoking cessation study, or it's a physical activity study intervention. People know what they're doing, and investigators may know what a person is doing. So maybe we should have that the outcome people are masked as to what the intervention assignment is. Many times the sponsor wants to distance itself, and so they don't want to know who's getting what, you know, collectively or individually. So nowadays we talk about who knows about what's happening, who's getting what, and who doesn't know. So we now, it's very rare. In some journals, they still use the term double blind, single blind, but such jargon is no longer used, okay? Those things are old school, and people now say, these people knew, these people did not know. And that's very clear what's has happened. The purpose of this, though, is to protect the study validity or integrity. It's the ability to compare the intervention arms. So we minimize the likelihood of biases that come from, you know, the patients knowing that I'm going to behave this way, or the staff knowing that they manage the patient differently, or if the outcomes assessment is people know, they may measure the outcomes differently or assess the outcomes differently. So in order to avoid potential biases, we have these systems. So these are essential systems. And if you're not able to have it, you have to justify why you're not able to have it. Okay. So when can it occur? It's actually throughout the study. It occurs at different places. It occurs at the process of randomization. So we don't uncover who is going to get what until necessary at the moment that they are assigned to it. So we call that the allocation concealment. During follow-up is what we traditionally understand as the blinding part. So it's of subjects, of care providers, of others. And then during analysis at the outcome assessment, the outcome assessors could be blinded, or the statistical analysts and the data safety monitoring board could be blinded. So as you're designing a trial, you must decide how you're going to blind and who you're going to blind, participants and or staff, and how you're going to do it. So if it's a drug intervention, it's one of the easiest things to do, if it's possible, to have the control arm intervention and the intervention arm to be the same, indistinguishable in color, shape, taste, smell, dosing schedule as well, packaging, and side effects. Because if one causes diarrhea and the other one doesn't, people will know what they're getting. Behavioral interventions is almost difficult or impossible to do. Okay, but clinicians and staff, it's usually harder to do, because since they are seeing the patient, they will know the side effects, they have access to the medical records, they see vital signs, and they are smart people. They will be able to figure out who's getting what in general. But it is advisable, to the extent possible, is that those that are administering the intervention, so those that are dispensing the drug, or those that are giving the counseling intervention, or those that are giving the physical activity intervention, they may be different from those that are managing the participant or the care of the participant, for example. So how do we conceal allocation? In the old days, we used to, once we have decided the randomization scheme, we would create these envelopes that were opaque, sealed, and sequentially numbered. And then they would take the next one, and then, the moment open the envelope and look into it, you had to, there was one way of doing it. Nowadays, we have numbered and coded drug containers or pill bottles. It's usually done centrally. It's not done at a given site if it's a multi-center study. And we also, most of the times, we now don't even create it on paper. We have it on a computer in an encrypted schedule. And that's one way of doing it. Another way of doing it is create it on the spot. So we don't have it previously done. We randomize at the moment. We have an algorithm that's running the computer that creates it on the spot. So that's another way of doing it. We'll talk more a little bit about the randomization in a few minutes. So we always want to make sure that the allocation was concealed. And how do we know it wasn't violated? So one thing to do is to assess comparability at baseline of what? Of the two groups, the usual care and the intervention arm. And here's an example of a small study of 150 or so subjects that were randomized to intervention or usual care. And we look at various characteristics. And we want to, for check and allocation concealment, you would expect that the differences between these were similar. And so this was a study on cardiovascular and heart disease risk. And so one of the baseline characteristics was a 10-year cardiovascular or coronary heart disease risk. And we noticed that there was a difference between them, statistically 0.03. Is this something that maybe the people that had higher risk were being assigned to usual care, the control arm, and people in the intervention arm have lower coronary heart disease risk? So now this is something that may make you be suspicious. Somebody may have been cheating. Maybe those envelopes were not really opaque, and people would put them up to the light and they would see, aha, and then they would change what happened. So you always need to check this, right? We never know if it's true or not. How do you mask participants? As I said, for drugs, it's very easy, and behavioral interventions, it's difficult to impossible to do. Masking staff and clinicians, as I said, is very difficult because they have access to a lot of information, and we need to just keep people separate. Here's an example of side effects likely to unmask staff and clinicians. So it's a study for treatment of common cold and receiving zinc or placebo lozenges. And we noticed that this is, again, a small study, so you expect that there would be some differences, but here we see that there's nausea caused by the zinc, and we have also that there is bad taste caused by the zinc. So if somebody knows this, then they say, oh, if I have nausea or I have a bad taste in my mouth, I'm probably getting the zinc. And so it potentially is unmasked, okay? If you don't have these differences, you could still check if masking was successful by asking. At the end of the study, you ask people, and people do this sorts of things, they ask the participants, the physicians, the clinic coordinators, what do you think this person was taking? Was they assigned to intervention or were they assigned to placebo? And you ask the patients individually, what do you think you were assigned to? And you can tell that this is the percent of participants who guessed correctly. And so the patients were 80% of them guessed correctly and they were getting propranolol, but placebo only 57. You notice that physicians are better at guessing who's getting placebo and who's getting propranolol, and the clinic coordinators as well. So the patients are not so well if they're taking placebo, but clinic coordinators and physicians, as I said, the healthcare providers have so much other information, they can guess a little bit better. What is the problem with guessing is that then their biases, if they had guessed already and during the study, then their biases may have crept in. So this is something that we usually worry about. So in many studies, it is impossible to mask during the study because of the type of intervention or the side effects, etc. So one thing that we tend to try to do is that we have the outcome be a hard outcome. So it has to be maybe a laboratory determination. So if your glucose is above this or below that, there is no uncertainty there of biases creeping in the measurement of whether it's a laboratory value is above or below something. Or it could be something as a hard endpoint like a death or a heart attack or a stroke determined by MRI or something like that. So if it's objectively measured, we call those hard outcomes. But another way is that the assessors of the outcome are not the people that did the patient care or the manager or administered the intervention. So they're external people. So you hire a group of external investigators or experts and clinicians that now look at all the documents and decide, yes, this person had the outcome, or no, this person did not. This is possible to do, takes effort, takes a lot of money. It's usually used quite a bit in heart disease and cardiovascular disease studies. There's a good example of one in multiple sclerosis that I was involved with. And they asked me, can you check if masking actually occurred here? And this was, what we did is we had two different investigators who were masked to assess the outcome of multiple sclerosis. And unfortunately, they were investigators in the study. So they were not external, so that they actually knew the patients. And they, even though they got information that was masked as to who it was, they scratched out the names and the addresses, etc. they were given their own patients to say, tell me if you were certain, probable, possible, or doubtful MS for this person. And so the patients of rater A, when we noticed that rater A is rating certain, probable, possible, and doubtful, and rater B is likely to be more probable and certain, then they are possible and doubtful. So there's he or she gives higher percentage of certain and probable than rater A, looking at rater A patients. But when they looked at rater B patients, rater B was even more certain. So this is higher here. And so even though they masked, they were not really masked, right? So this is what we try to check if people are really masked on this. So to summarize on masking, it's a desirable design feature because it helps you avoid certain biases. It protects the study from those biases, so you are now more objective. It's not always possible, and it's always not necessarily, when tried, successful. But one thing we do is we avoid use of this jargon of single blind or double blind, but and specify who was kept from the knowledge of the treatment assignment and how. All right, so now we want to move on to another ethical issue, and that is the randomization. In observational studies, we take a random sample. In an experimental study like trials, we have a population of interest. We define inclusion and exclusion criteria that determines a target population. And we do not take a random sample of the target population, but rather we select a non-random convenience sample, people that come to our clinics, and self-selected volunteers that agree to participate. So consenting eligibles is a non-random convenience sample. Every clinical trial is a non-random convenience sample. As statisticians, what do we call that? Garbage sample, okay? Why do we do this? For ethical reasons, right? And that's where randomization comes in. So if you hear that somebody said, we are comparing our intervention against historical controls, that is a garbage study. If they say that the researcher assigned people to the interventions, that's personal bias. If they have said we did systematic randomization so that, it was alternating, the first patient got control, the second one got intervention, the third control, intervention, etc. You alternate it. Or you say, the medical records or their ID number, if it ends in an odd number, they go to A, and if they end up with an even number, they go to B. Or those that come to me at my clinic on Monday, Wednesday, Friday go to A, and Tuesday, Thursday, Saturday go to B, or based on the date of birth. Any of these things as a criterion for assignment is predictable, as you can know beforehand. So then personal biases come in. So none of these things are good options. These are bad options to do non-random allocation. What is achieved by randomization? We want objective allocation. We want an equal or known probability of being offered each intervention. All prognostic variables, known and unknown, will have an equal distribution in the treatment groups in the long run. So you're now going to be protected from confounding. And treatment groups would on average have the same prognosis if it were not for the interventions. And then will allow us to test the statistical assumptions, okay? So what we're trying to achieve with randomization is fairness, right? So it's unbiased allocation. Notice that unbiased does not mean equal, because you may want to do it unequal allocation. So we either have an equal chance or you may want to do it unequal. And why do we do this? Well, we may want to help in recruitment by saying, you have a higher chance of getting into the intervention than the control. Well, then what happens with equipoise? Is equipoise really there? If we think that intervention is better than control, then why are we doing the study? So this becomes an ethical issue to do unequal size, but sometimes people do that. More commonly is to do equal size. Fairness is at least in the randomization process. We're trying to balance the equal numbers of people, but also the similarity of the subjects in each arm. So all prognostic variables, known and unknown, have an equal distribution in both arms, and on average have the same prognosis if it were not for the intervention. So we say this is no confounding. So if you look at the paper on the Pfizer vaccine for COVID, you have always at the reporting of a clinical trial, the famous table 1, which is the demographic characteristics of the participants in the main study population. Here, in this case, is the vaccine group versus the placebo, and then they have the total. Notice, though, that there is no p-values. Table 1 is comparing these two, and there is no p-values. It is now standard practice that this should never have p-values. Why does it not? Hey, Dr. Vasantha, please stay on mute. Why don't we have p-values? Well, because there's no hypothesis that you have beforehand that I want to check if I hypothesize that I'm going to get differences. So there is no hypothesis. And secondly, look at this. If I look at the male and female distribution, here I have 51.1, 50.1%, 48.9, 49.9. No meaningful differences. But because this is a huge study, it's statistically significant. So if I had put a p-value, because I did a large study, oh, there's statistical significant differences in sex. So therefore you have to adjust for sex. No, you look for meaningful differences, okay? And there's no meaningful differences. You look throughout there's no meaningful differences. And that's the objective here, okay? Now, what are we trying to achieve also with randomization? Remember that I said that we're using a garbage sample, right? Because it's a non-random selected, so self-selection, convenient sample. It's a garbage sample. So without randomization, we have this garbage sample of subject. Now, thanks to the principles of randomization tests designed by R.A. Fisher in the early 1900s, we have that he showed that randomization works in his experiments. He actually also had a very good development example of where the Fisher's exact test came from. And it was the story of one lady who said that she could distinguish whether milk was poured first in a cup of tea or tea was poured first in a cup of tea and then milk on top. And he devised a test based on randomizing the order in which things were sent to her to test whether she could tell the difference or not. And she actually was able to, but that gave origin to Fisher's exact test. So the concept of randomization tests and of randomizing people to different interventions is essential to the conduct of clinical trials. And that's why we call it RCT, is randomized controlled trials. The C is for control, not for clinical, because it applies to non-clinical studies as well. So how do we operationalize randomization? We can do it simple randomization by doing a coin toss, or we can do an urn with replacement, which is the same thing. But if you toss it, let's say you randomize it 50 people, you may get 26 and 24. And so you may get slight imbalances in treatment allocation, or you could get larger imbalances. You may have imbalanced sequences also. So you may have the first 7 are heads, and then you get tails. And eventually, if you do a lot, you may get it to be balanced, but you may have imbalanced sequences within that. And if there's temporal effects, this may affect things. So we tend to do some sort of restriction on the randomization. We don't do totally unrestricted. And so we do it like an earn without replacement. And those are fixed allocation schemes. May give some slight imbalances in covariates, but may have imbalanced sequences, although balanced in the end. So it's important if we have temporal trends to not even have imbalanced sequences. So in order to further correct that, we do blocking. And you may have heard these terms. Let's say we have blocks of 4 subjects. So then if you have A and B, we have 6 possible block sequences. So you have like AABB all the way to BBAA. And so what you do is you develop the randomization sequence by randomly selecting one of the 6 possibilities with replacement every time. So you maybe take a dice, toss it, number 5 comes, so then you select this. Then you toss it again and you get number 2, you select this, and then you start building up your sequence that way. Now, what blocking achieves is numerical balance, statistical efficiency, the appearance of success, and you avoid the bias of secular trends, right? And balance of near balance if recruitment is stopped prematurely. A further safeguard that we do against allocation bias is that if somebody, like the investigators are some very clever, and they know the block size and the previous allocations, and they know the side effects, and they say, oh, wait a second, the first 2 got the intervention, or the first 2 were having side effects like this, so the next 2 are going to be getting this. So if we now change the block size in random order, it makes it harder for them to guess, right? So now, in order for investigators not to guess, we do what's called permuted block randomization. We first choose different block sizes, and maybe like, for example, 2, 4, and 6. So now we have, these are the possible blocks that we can have. There's 20 here, there's 6 here, there's 2 here. And the order in which these we take them is random. So the first we may get one of these blocks, and then we may get this block, and then we may get this block, and then we may get this, and then this, and then this, etc. So it's very, very hard for them to guess, even if they know side effects and they're trying to guess, because they're all humans too. Nobody likes secrets kept from them. They're going to try to guess. Okay. So this will now get the balancing of the numbers. It'll get the balancing of the sequences. But what about covariate balances? How do we handle covariate imbalance? The best way is large sample sizes. As you saw with the vaccine study, when you have large sample sizes, everything is balanced, okay? But there are other ways of doing it. There's stratified randomization and things called minimization. What are those? Well, stratified randomization is, if I think I want to make sure I have balance on, let's say, age groups. I don't want older people in one than in the other. And I have age group 1, age group 2, age group 3. I first get the eligible subjects, I classify them as to their stratum value, and I allocate within the stratum. So now when I get the control arm here, I will have the same distribution of age groups as I'll have in the intervention arm here, okay? So when do we stratify? Only if you have a few baseline characteristics that are strongly related to the outcome. Right? If you have many of them, how many strata can you handle? Not usually easily. It's usually done in small trials to prevent imbalance on major predictors, because if I have a large numbers, everything will be balanced, okay? We especially do it in multi-center trials. The stratum becomes the center, because even though we standardize the procedures and we train people, they don't always follow things exactly like that. So there may be center effects. So in order to distribute the center effects equally, among the intervention arms, we always do stratified randomization by center. Okay. Why do we not do stratified analysis instead? Yes, it's better to prevent than to try to treat, right? So that's why, observational studies, we do stratified analysis because we are not able to stratify beforehand, right? And so here we are able to, so to the extent possible, it's best to do it. If you have many small strata, we have like match randomization, for example, in randomizing twins or family members. You can have families, and within a family you randomize, or within family pairs, you can do it like that. Okay, this is very restricted randomization. Okay. Now, sometimes individual randomization is not possible. And so the intervention may be an educational one, or maybe a physical activity one, or some involves counseling or behaviors by the staff. And so there was potential of contamination of the intervention arms. And interventions have to be provided because of the nature of the intervention in group settings. I'm going to do it in children that are school-age children, or I'm going to do it in factory workers, or in churches. Or it may be unethical or impossible to single out subjects. So if I'm doing a community-based intervention, I cannot say, oh, wait a second, we're going to do a treatment of the water supply. How can I keep some people from drinking the water and others not drinking the water? So you cannot do environmental change interventions are always grouped interventions. So we call them, how do we randomize? We randomize clusters, or we randomize groups of individuals. So now what we have is let's say, clinics, and if the contamination is, the staff have to counsel people. So now you cannot counsel one person one way and not contaminate counseling another person a different way. So we have you counsel only one way. So we now have everybody in a clinic counsels with the intervention counseling, and everybody in other clinics counsels with the control counseling. So now clinics are randomly assigned, and I have fewer units being assigned. But everybody that goes here, all the patients here get the intervention, all the patients here get the control. Okay. So now the unit of randomization is the clinic, but the unit of analysis continues to be the patients or subjects. Okay. And so patients within a clinic, their outcome is correlated due to what we could call, in parentheses, clinic effect, right? And the variance both within and across clinics needs to be accounted for. Okay. So we take into account that into the sample size. And Nisha, Matthew, please. Randomization, though, because we're randomizing a few units, it's unlikely to balance the arms. Okay. And therefore, we have to do something else to covariate balance, right? How do we do it? One method proposed is called minimizations. If we have a few subjects and we know a few important covariates that we want to make sure that they are not imbalanced, okay? We need to be able to measure those and know it. So at the group level, we know characteristics of the groups or of the clusters of the clinics. Then we try to do it. How do we do this? So if I have these subjects, N subjects, and I want to allocate half of them to one intervention or the other, I can calculate all the enormously large M number of possible random allocations of those N subjects. Then I will develop a discrepancy index that's based on these M. Oh, I should use a different one here. This should have been K. So that's my mistake. But if I have prognostic factors, now I develop a discrepancy index, and I calculate then the all the different discrepancy indices for all of the possible random allocations. and I select just the one that is the smallest one. So that's the one that will give me the least discrepancy between the arms on the sets of covariates. The problem with this is that since you're selecting one, it was seen as deterministic and not random, and nobody uses minimization. So an alternative to minimization is called optimized randomization. The first three steps are exactly the same, but now we create a subset of K tolerable allocations out of all of these M allocations based on them having a discrepancy index less than some threshold that you make up. And then now that you have these K, you choose one allocation randomly among the set of K tolerable allocations. And because you have randomly chosen one, then it's called optimized randomization. You use the word randomization, and it's acceptable. This is a study that was done that's a good example of the use of this. And they actually have the article on the trial is there, but they also have an article that describes the method of optimized randomization as well. Other techniques that are done for assigning people are adaptive randomization designs. These are using previous information in the study to then choose where does the next subject go to. It's most often used in phase 2 trials. Rarely, if ever, do you see it in large trials, right? So for example, we have a bias coin that's allocation adaptive. So if the first patient comes, they have a 50-50 chance, but if it's randomized to A, the next patient now has higher probability to go to B. And let's say this person still goes to A, then the third patient has even higher probability to go to B. And now this one goes to B. So now the probability to go to A is increased a little bit. And you just keep going. So once we have it matched, now this goes back to 1/2. And so if this one goes to B, what is the probability here? It's going to be 4 7s, 3 7s. So you increase it here, etc. So this is called a bias coin, and it's adapting the randomization to who has been allocated to what. There's another one called play the winner. That this is now response adaptive. And that is you assign the first patient randomly, and let's say they get A and they get a positive result. The next one then is assigned A because A was positive. But when A turns out to have a negative result, now the next one goes to B. If B is positive, it goes to like that, and it just keeps going, and you're playing the winner as you go along there. So at the end of the study, you always want to check was randomization was successful because maybe some people have cheated, it did not go well, or you're just unlucky. How do you check? You review that table 1, but for meaningful differences, not based on p-values necessarily. People like to do that, but you want to compare the treatment arms at baseline. Large trials will almost always have simple randomization work, and in small studies, almost always simple randomization will not work, okay? So you want to check the balance at the end. So here's an example of, this was a study with 1,000 Nepalese children, a subset of a larger study on vitamin supplementation on growth. And they wanted to make sure that the mother's age was balanced between the intervention arms. So I created some hypothetical, simple random allocations, 4 different ones, just to illustrate this. In allocation 1, if I have 1,000 subjects, I got the mother's age mean between the arms to be fairly comparable. In some simulations I did. If I had 500, there is some differences, 29.7 and 29. So, but it's fairly comparable, so negligible. In very small now, so I have 100. In this case, I just got bad luck in this simulation, and it ended up that there was significant difference. Or I could get good luck, and I still get it. So the chances of getting good or bad luck are, the chances of getting bad luck are higher it's smaller sample sizes. The larger the sample sizes, things are likely to work. Okay. So if the randomization is set beforehand, we call it fixed. If it's changing with the next person, we call it adaptive. But if it's fixed, you have to conceal it, right? So that people do not know what the next one is. But if we do not create it beforehand, we can be adaptive with either the bias coin, play the winner, or sometimes we want to make sure that the covariates are adaptively balanced. You now do it with regression models. It's a slightly more complex bias coin. And concealing it, as I said, is it's done by preventing interference with the design and preserving the objectivity. Okay. The final thing I want to talk to you about, and I only have 5 minutes left, is on the needs for external monitoring. And we're going to go more into detail about this tomorrow. But the rationale for monitoring and conducting interim analyses is driven by ethics. Ethics dictate the need for considering the possible early termination of a clinical trial based on analytic considerations of that you have more toxicity in one, or you have evidence already of efficacy, or do you have zero tendency, or you have administrative reasons like, the study is just being done so poorly, why are we wasting our time? Or there's subjective reasons that things from other studies have studied this and shown that this is not working, or it's working, or there's, the practice, this is no longer a viable alternative. A new drug came out that's much better than this, so why are you doing this? So there's analytic, administrative, and subjective reasons for it. There's always an external data and safety monitoring board, not internal. Nobody from the study is a member of that board. They have meetings with different purposes throughout the study. They have some during the recruitment phase, some during the intervention, and during the follow-up phase. They are usually equally spaced, but maybe towards the end, they're closer together. They're scheduled by the sponsor, and they have different purposes. It's a board that's advisory to the sponsor, external, with no conflict of interest, with a different amounts of expertise from different people, usually a small group, an odd number of members. The roles of them is to monitor the integrity of the data and the study, and the individual safety of each subject and the collective safety of each subject. So therefore it is not data monitoring board, it's data and safety monitoring board. So they're monitoring the data, they're monitoring the safety. So the data means the completeness, the timeliness, the quality, accuracy, all the processes about it. They monitor the integrity of the study progress, this recruitment, follow-up, all the adherence to protocol, etc., and the safety of the patients individually and collectively. So they have a big responsibility. Their role is they're responsible for evaluation of equipoise. And if equipoise was enabling you to start the study, lack of equipoise during the study is a consideration for early termination. It only makes recommendations to the sponsor, the team of investigators. And how do they actually do the monitoring? They have periodic meetings during the conduct. There's an open section where they look at the study integrity, and everybody participates. The study investigators come and report on the quality and the study progress. Those people are invited to leave. And then there's a closed section where now they're looking at the adverse events, they're looking at the outcomes, and the DSMB decides if the study has reached a conclusive answer, should it proceed or be stopped? Or also, if the study will never reach a conclusive answer for whatever reason, should it proceed or be stopped? Okay, so I will stop here. The next lecture, we will go into the details more of analysis of an RCT. We'll be talking of the types of analysis that is done at the end of the study versus during the study, what the DSMB does, what is the statistical analysis plan, who do we study? We'll talk about the different analysis populations and the types of analysis. What is primary? What is secondary? We'll focus also on problems in analysis, like missing data. How do you analyze change? And specifically, interim analysis. We will also conclude with a necessary sample size and calculations related to that. So I thank you, and I apologize that I don't have much time for questions, but we can take a few minutes for any burning questions that people may have. Thank you.
Hi, sir. I'm John here from St. John's Chicago. So my question is regarding what could be a suggestion on randomizing for the studies, especially in surgical studies, where there is no blinding is possible, and always there is the specialization, especially in neurosurgeries, the doctors are very less. So the outcome assessor and the randomizer will be the same, and the bias is more possible. And whatever the allocation we give, but at the table, the doctor knows what surgery he is going to give, and the patient also know what is the surgery he has undergone. So could you suggest still to go with the randomization process or to go with other non-random allocation?
Well, no, first of all, non-random allocation is worthless. You should not ever consider not randomizing. Now, if it's the subject has to consent to being randomized, and the physician needs to say that it is correct that the two types of surgeries are equally positioned, and so either one of those is okay for this patient. If the physician thinks that this person is, or this intervention is better for this person, then that patient is not possible to be included in the study. You just treat them with whatever you think is the best thing for that person, because they're not equal poised. You cannot do it. So that particular patient becomes ineligible. If you think it's okay, either surgery is okay for this patient, but the patient says, no, I don't want it. I want this one. I don't want that one. Then the patient is not willing to consent to being randomized. Then the patient is not in the study. So for that particular patient, they may not be in the study. So it is always, you have to take the patient's viewpoint and the physician's viewpoint here. Now, if the two interventions being done in the surgical study is the clinicians are convinced these two methods, there is no evidence to say currently that there's any difference, and for this patient it's okay, then that's a study that can be done. And now you have to get patients for which those two interventions are equally likely to benefit them, or maybe not, and they consent. Then it's possible to do. Now, as far as the randomization process, what usually is done is that when the consent is done, right when the patient is going to be put under for the surgery, at that moment, you click on the computer or open the envelope and say, I'm going to do procedure A versus procedure B. And so now they know what they've done. And assuming that you have to assume that the surgeon is going to do the procedures equally well. They're not going to, I want procedure A to work better than B, so if I have to do B, I'm going to do it poorly. That's unethical clinically. That's out of the question, right? They should be ethically doing the intervention. And the outcome should be assessed by a colleague somewhere else that's not part of the study. I find it hard to believe that in India you won't have another neurosurgeon that can at least look at the outcome. That you cannot find somebody like that, right? So you are able to find people. You may not want to have to pay them to do this, but it's advisable unless the outcome is it's an image in the MRI or it's something that is a radiologist could look at or somebody else can look at. There are usually ways to find somebody that can do this outcome assessment objectively. It can either be an objective outcome or you can find somebody that can determine it objectively. So, but again, the answer to your question is you should not consider never randomizing. You should always consider randomizing if you want your study to be believed.
Yeah, thank you, sir. And one more question. This is one study which I recently come across for a randomization. But the ethical part is not clear for me, where I am not able to answer whether it is ethically right. That is, a surgeon is going to do a surgery and he is going to decide what surgery can be done based on the MRI as well as the X-ray. So he says the new MRI techniques gives the additional information and the practice is doing by X-ray. So he wants to randomize deciding on treatment plan based on X-ray versus MRI. So now, is it ethically right to decide only using X-ray now?
So
he came out with a paper from China. They have written a protocol paper. So they have did the same study for doing X-ray and MRI. So I'm not able to say no there, but I'm still confused whether that is right or not.
So in this case, the intervention is not the actual procedure. The intervention here is the source of information. So you're randomizing which source of information can I use, so that's the intervention. In this case, is which source do I use? Who decides whether it's ethical is not me or a statistician. It is an ethics board. You need to ask that of an ethics board, and I will not take a stand on this because I don't know enough about the considerations that would come into place with this type of intervention. But I would consult your internal institutional review board, and they will tell you if it's ethical or not. And if they say no, then you can't do it.
Thank you, sir.
Question from Kalai. Kalai?
Sir, good evening. This is Kalaivani.
Hello?
So yes, every study needs allocation concealment. Let's say that the intervention is packed and reached the study center. Still, we need allocation concealment.
What do you mean by a study is far away? You mean you have to, is it a drug study? It's a behavioral study? What kind of a study is it?
So let's say that it is a drug study. As you rightly mentioned, that it is indistinguishable by color, taste, smell, and so on. So it is packed from the manufacturing industry itself and reached the study center.
Okay.
Still, allocation concealment is necessary.
Well, so how are you randomizing there? Each center now has a bunch of packets that are numerical, and that some of them have the active and some of them have the control, and they're in sequence there, and so you've already concealed it in that, right? So that's what we mean by allocation concealment. It's just instead of the physical envelopes, you have packets now that are the envelopes with the drugs inside. So that's what concealment is in that case.
Thank you, sir. So one more question. Can the study statistician be there in the DSMB meeting?
Right, so we'll talk more about it tomorrow, but yes, in the open session,
You will be there.
study statistician is just like another study participant. All the investigators and the study statistician is in the open session. All of them are. In the closed session of the DSMB, there is what we call the independent reporting statistician. So the independent reporting statistician is the statistician that knows the unblinded results and does the calculations for the unblinded results. So if the study is being done out of one center, one institution, and you don't have the statistician of the study be the same as a reporting statistician to the DSMB. You have to have at least two statisticians. So if you have the study statistician remains blinded during the study conduct, and you have an unblinded statistician. However, maybe you don't have the resources of having two statisticians. If you have only one statistician, that person has to do the unblinded analysis. So if they are unblinded, their interactions with the investigators should be minimal to none during the study conduct, because they already know the results. And let's say the physician say, oh, we need to consider whether we need to adapt the sample size because maybe we don't have a big enough effect going on, or the effect is smaller than we thought. The statistician knows he or she cannot be involved in that discussion, right? And so that's why we want to keep them separate. So the statistician that is unblinded to the study results during the conduct of the study should not be involved with any decisions of the study management, study conduct, etc. So that is very important to keep them separate. So maybe some people can do it. One person could, keep a poker face and not smile or do anything, but others cannot, right? So you want to keep people separate. Ideally, that's why the statisticians that report to a data safety monitoring board are statisticians at a contract research organization, or they're statisticians at a separate coordinating center, but not statisticians that are actually involved in the study conduct.
There's one more
Thank you,
question
sir.
from Tinko. Yeah, one more question from Tinko Thomas.
Thank you, Dr. Bangdiwala. My question is with regard to the data management. With recent advances in devices being used in diagnosis or any of that, and artificial intelligence, which is beyond the scope of us statisticians, my experience has been that the data is actually being generated by the people who develop the device, or actually, so there is a conflict of interest. And what my experience is, I often feel very uncomfortable in handling that data because I'm not sure about how unbiased that data is. Do you have any such experience, and what would be your recommendation, if so, to make sure that the data that we are handling is true, I would say?
Yeah, no, that's a very valid point and question. I have not had that experience of working with device people, but in general, we statisticians are held responsible for the data and the quality of the data, but we are not the ones that are generating the data, right? So when we have to be trusting and that whoever is collecting the data on paper forms, for example, at the clinics is doing it correctly, or that whoever is generating the questionnaires or who is filling out the pro forma at different places, they're doing it correctly. So we depend on the clinical staff to do it correctly. So if they don't do it correctly, the data are going to be wrong. So they'll take it to devices. If the devices are not generating the data properly, we still have to trust it, right? So how do we trust it? How do we trust the people in the clinic staff? Well, we monitor them. We periodically go and check on them. We do all sorts of quality checks. We should do the same thing with the device data. We should ask the developers of the device to demonstrate to us that the device is actually collecting the data accurately. And so they must have, in order to have their device approved by some regulatory agency, that it is, monitoring heart rate properly, that it's collecting glucose properly, that it's collecting whatever it is, movement or whatever it is that you're collecting on the device, that it's collecting it properly. So they have to have gone through quality control processes, and somebody's regulating them. And so if I'm doing the study, I will not get the device from company A that is unregulated. I will get it from company B that has proven to me that they've done it properly. So you can demand this. The study investigators will definitely want to demand this, because the integrity of the study depends on the quality of the data. And if you cannot trust the data or the people collecting it or the device producing it, that's a serious problem.
Thank you.
So yeah, so I would demand them to produce evidence.
Thank you.
All right, I think we're 15 minutes past the time, so I will
Okay.
appreciate
Thank you
leaving
very
now,
much. Yeah.
and we'll see you tomorrow. We'll focus more on analysis tomorrow.
Okay. Thank you very much, Kant.
Okay, thank you.
Okay. Bye.